Organisms show variety in cell number, shape and size. So we are learning under separate headings how the organisms are having different number of cells, have different shapes and size of cells. So here you are looking at the diagrams of amoeba, paramecium. Here this represents the algae having multicellular cells, which is called a Volvox colony. Here you are seeing different shapes of the cells, where you are seeing red blood cells, muscle cells and nerve cell. Here is a osteocyte, which is a bone cell. Let us learn the different parts of the cell. So the three main parts of the cell includes the cell membrane, nucleus and the cytoplasm. Here the cell membrane, as you can see, the inner layer, this is the cell membrane. Then inside that you have the nucleus, and here the jelly-like substance called cytoplasm. So these three parts, the cell membrane, then the nucleus and the cytoplasm forms the three main parts of the cell. Here you are looking at a plant cell. So this is the ultrastructure of the cell. So you must be thinking, why we have to study the ultrastructure of the cell. So for that, what I can say is, so you have learned about the living organisms. So what consists of those living organisms? No doubt you know that those living organisms are made up of cells. Here when you look at the living organisms, so you will notice different parts or organs of those living organisms. Suppose in plants, you find the parts like the leaf, root and branches. So shoot system consists of leaves and branches, whereas you observe the root system. So when you are observing the leaves of the plants, so they do a very important function like photosynthesis. So the prepared food will be stored in the plant cells. Here you can see the chloroplasts, which are very large in number. They will helping in the preparation of the food, and the stored food gets distributed to all the parts of the plant with the help of a tissue. So the tissue is phloem. So group of certain plant cells form a specialized tissue called phloem. So with the help of that, the food gets distributed. Likewise, when you observe the roots of the plants, so roots will absorb the water, and that water will be supplied to all the parts of the plant with the help of again a special type of tissue called xylem. So where the xylem again consists of group of cells. So those cells help in the transportation of the water to all the parts of the plant. So when you are learning all these things, you have to learn about the detailed structure of the cell. So here this ultrastructure is only visible under advanced microscopes. But under an ordinary microscope, so when you want to observe the cells, you have to take minute parts of the plants. Here in your textbook, they have suggested the plants like Tradescantia, Elodia, or Rio. So you take the leaves of those plants and remove the very thin layer over the leaf and observe under the microscope. And then you will see the cells of the plant like this. And they have also suggested to observe the onion peels. So you take an onion bulb, remove the pink dry layers of the onion bulb, and after that you will observe white fleshy layer over the onion. You carefully remove that thin white layer and place it on the glass slide. Add 1 or 2 drops of water to that, and with the help of a needle or forceps, you pierce it into smaller pieces, and to observe the parts under parts of the cells will take up the different colors and they will be looking to the eye in a very bright manner. So likewise, you can study the ultrastructure of the cell under the advanced microscope, and you can observe these cells under the ordinary microscope as it can be seen here. So here the cells are seen here. So which is the cell membrane and which is the cell wall? Here you can see a layer. So that is the outermost layer. So this is cell wall. So this is cell wall. So next to that you find a cell membrane. So this is a plant cell. So plant cell consists of both cell wall and cell membrane. So here you can see the cell membrane here. This is cell membrane, and next to that you find a very thick cell wall. See, what is the function of this cell membrane and cell wall? See, as we have learned earlier, the cell membrane helps in giving different shapes to the cells. As we have learned in the blood cell, it is the red blood cell, and in the nervous tissue we learned the shape of the neuron or nerve cell, and we learned the muscle cells which are spindle shaped. So different shapes of the cells is because of the cell membrane. So a cell membrane allows the substances to move inside the plant cell or to go outside the cell, whether it is plant or later we study the animal cell. Here cell membrane allows substances to move inside the cell as well as to go outside the cell. And what about the cell wall? So why we find the cell wall only in the plant cell, whereas the animal cells do not have cell membrane? See, the cell wall is a very thick membrane. So this cell wall is made up of certain chemical substances like hemicellulose, pectin, like that. That cell wall protects the plants against some of the drastic or some of the odd conditions of the weather. So in weather we find variations in the temperature, and high wind speed will be there in the weather, and atmospheric pressure also will be there in the climatic conditions. So to face all those things, plant need to have a protective wall, so that is called cell wall. See, as the animals, so they get protected inside the shelters like the house or the caves, but here plants have to remain all the time in the harsh environmental conditions like the high temperature or the high wind speed or the atmospheric pressure. So they have to have a strong covering, that is the cell wall. So this cell wall can be observed under the microscope. So when we take that onion peel and observe, we will learn how onion peeling is observed under microscope. Take the onion bulb, water and dye in different watch glasses. You take water in another watch glass. Take the onion bulb, remove one of its top layer. You can see white fleshy layer inside the onion peeling. Carefully remove that white layer with the help of a forceps. You can easily remove it from that onion layer. So this white fleshy layer is dipped inside the water. So dipped inside the water which is there on the watch glass, and from that you carefully take a piece of that and immerse it in the dye, so which is orange in color. After a while, you remove that specimen or the onion peelings from it and again immerse it in water so that excess dye gets removed. Take that onion peelings from that water and place it on a glass slide. Add a drop of glycerin to that so that no overlapping of the cell takes place. Then carefully place a cover slip over that. You see that no air bubbles get entered into the cover slip. So to remove the excess amount of water around the cover slip, take a tissue paper and wipe it around that. So the slide is ready to observe under the microscope. You can observe the cells of the plants. So the onion peelings have the plant cells. So here you can clearly see the cells which are separated from each other by distinctive cell membrane and cell wall. You can see the dot-like structures. Those are nothing but the nucleus present inside the cell. You can see the epidermis. Under the heading Cell Structure and Function, we are learning parts of the cell where we are learning cell wall. Here you can see the plant cell. So plant cell, a part of that get enlarged to see the cell wall distinctively. You can see the parts of the cell. Plant cell, that is vacuole, nucleus, cell wall, cytoplasm, and you can see the innumerable number of chloroplasts inside the cell. So when you observe the onion peels, the cells will be looking like this. So which shows the thick cell wall, and next to that so is a cell membrane. And while studying the animal cell, so this is the ultrastructure of animal cell, where the parts you can see are cell membrane, vacuole, nucleus and cytoplasm. So inside that you find a jelly-like substance called cytoplasm. So to observe animal cells under the ordinary microscope, human cheek cells are used. So where you can see number of cells getting divided by the cell membrane. Here the cell wall is absent in the animal cell. So we learned that in the plant cell, so there is a cell wall which gives protection against the harsh environmental conditions like the high temperature, high pressure or high wind speed. So it also has got cell membrane which gives various shapes to the cells. So here you can observe the animal cell which is having only the cell membrane and the cell wall is absent here. So this is the ultrastructure of the animal cell, where you are observing the cell membrane, vacuole, nucleus and cytoplasm. So as I said, so there are three main parts of the cell, so consisting of the cell membrane, nucleus and the cytoplasm. Here cell membrane is a porous layer, it is also called as plasma membrane. So it is a porous layer which allows the substances to move inside and outside of the cell. So this cell membrane separates one cell from the another cell and also separate the cell to its outer medium. So here the medium will be having the different substances and the cell will be separated from that outside medium by this cell membrane. Here you find a nucleus which is a dark spherical shaped structure, and you find a jelly-like substance so outside the nucleus and inside the cell membrane. So this is called the cytoplasm, which consists of various cell organelles. So that you will be studying later. To observe the animal cells, so under an ordinary microscope, have to take the human cheek cells. So cheek is the part of the face of the human beings. So inside the cheek you find a mucous membrane, so which consists of a whitish layer over that. So to remove that white layer, you take a matchstick. See that it is not sharp, make it blunt, and carefully you scrape the inside of the cheek and drop, and you take that whitish layer and place on a slide and add a drop of water to that specimen you have taken from the cheek. So that whitish layer will be placed on that slide and it gets distributed when you put water to that, and take a needle and spread those specimens across the drop of the water. To make the cells distinctively visible, you add a drop of iodine, or you can also put a stain like methylene blue. So when you add a dye, so the parts of the cell will take up that stain and they will be visible under the microscope very brightly. And you can see the cells under the microscope which gets separated from each other by the distinctive cell membrane. Prepare a slide consisting of human cheek cells. For that you have to remove the inner lining of the mouth, so consisting of cheek cells, with the help of a blunt stick. So you remove carefully that whitish layer present inside the mouth. The inner lining of the cheek consists of the squamous epithelial cells. You smear that specimen taken from the mouth onto the glass slide. Place a drop of methylene blue solution which is diluted and place a cover slip over that. Care should be taken that no air bubbles enter into the cover slip. Remove the excess of stain coming out of the cover slip with the help of a tissue paper. You gently tap with the help of a needle from its backside and wipe the excess amount of the stain which is there around the cover slip. And the slide is ready to observe under the microscope. You can see the triangular shaped or polygonal shaped squamous epithelial cells which are there inside the cheek cells of the human beings. So here the animal cell is considered as eukaryotic cell as it has got a definite nucleus bounded by the nuclear membrane, whereas here you are looking at a bacterial cell which is considered as prokaryotic cell. So where you have nucleoid region, the nuclear region is not bounded by a definite nuclear membrane. So we will learn about other parts of the cell. So far we have learned about the cell wall and cell membrane. So let us learn about the cytoplasm and the nucleus. Here you are seeing the animal cell. So inside that you have a jelly-like substance. So you have cell membrane and the nucleus. So in between the cell membrane and the nucleus you find a jelly-like substance called cytoplasm. See here, cytoplasm is different from protoplasm. See, what is protoplasm? So to know that, we have to know about the nucleus. So inside the nucleus you find a spherical dark shaped structure called nucleolus. And you find here also a substance called nucleoplasm. So outside the nucleus you have cytoplasm, whereas inside the nucleus you have nucleoplasm. See, both the cytoplasm and nucleoplasm together forms the protoplasm. So hope you have understood about this. So the cytoplasm, what it consists of? It consists of various cell organelles. See, cell organelles. So can you name the different cell organelles which are present inside the cell? So you have learned all of them in your 7th standard. So here there is a list of all the cell organelles present inside the cytoplasm. So we learned about the cell wall, cell membrane. So cell wall is exclusively present in plant cell. Here cell membrane is also present in plant cell, whereas both cell wall and cell membrane are present in plant cell, whereas only cell membrane is present in animal cell. You have cytoplasm, nucleus, Golgi complex, endoplasmic reticulum, chloroplast, vacuole, ribosome, lysosome, centriole and mitochondria. So here I have listed 12 different parts of the cell. So inside the cytoplasm you find the various types of cell organelles. See, this cytoplasm is the center of all the cellular activities. And since it consists of different types of cell organelles, each cell organelle will be performing its own specific function. So after the cytoplasm, so we will know something about the nucleus. See what this nucleus consists of. Nucleus is surrounded by a nuclear membrane. See, as we have learned about the cell membrane, see this nuclear membrane is also a porous membrane. See, this nuclear membrane allows the substances from the cytoplasm to get inside, and some of the substances inside the nucleus allows them to go outside. So this nuclear membrane is present in certain type of cells, that is called eukaryotic cell, whereas this nuclear membrane is absent in some of the cells like prokaryotic cell. So here you are seeing the bacterial cell. So we will be learning that in later. So nucleus has nucleoplasm in it and is surrounded by the nuclear membrane. So this round, small, spherical structure is called nucleolus. And you see these structures. So these thread-like structures. So what do you call these thread-like structures? So this is nothing but the genetic material or nuclear material. See, what is this nuclear material or genetic material? So this consists of the hereditary substance called chromosomes. See, inside the nucleus, so the thread-like substances will be converted into chromosomes. You are looking at this butterfly-like structure. So these are very ultrascopic structures. So which are seen only under advanced microscopes like the electron microscope, but this can be seen under the ordinary microscope also, so like these structures. So these chromosomes are the hereditary substances. So when you carefully remove the substances from it, so it consists of thread-like structures called DNA. So DNA is nothing but the genetic material. So the long form of DNA is deoxyribonucleic acid. Deoxyribonucleic acid. So inside the nucleus we have genetic material which consists of chromosomes. So chromosomes in turn consist of DNA, so which is having the name deoxyribonucleic acids. So these DNA are double helical structures. So when ultrascopic structure of a DNA is learned, you see a helical structure all born to your parents, and people say that you are looking similar to that of your parent. So whatever characters present in your father or mother will be transferred to you. So by the means of these structures, so these are called genes. So unit of inheritance. See how these genes present inside the nucleus get transferred. See how an organism is born. Organisms are born with the help of their parents. So the reproductory cell of the father and the reproductory cell of mother gets fused. So while the fusion, so this nucleus also gets fused. See here, the one cell having the nucleus, having the genes inside that. So another cell, so having the genes inside that. So when these gets united, so both the genetic material gets united. And those characters will be present in those genes. So those genes will be coding for particular characters in an organism. Suppose you have brown eyes. So how you get those brown eyes? Because your father has got the brown eyes. Suppose you have curly hairs. See that you might think your mother is having curly hair, so that character is transferred to you. And you also see some of the different characters, like when those characters may be black or brown eyes, straight hairs or curly hairs, they get combined together to form another character in the next generation. Children may get some of the different combinations of these characters. So all these are because of the inheritary substances called genes. So these genetic material can be learned later in your studies. So the nucleus is the center of all the activities, as it consists of genes as the inherited or hereditary substances, so which helps in transfer of characters from parents to their offspring. So like this you learn about the nucleus, which consists of genes, and you learn about the cytoplasm, which gives space to various cell organelles. Here they have mentioned about two different types of cells. Those are called eukaryotic cells and prokaryotic cells. So what is the difference between these two? Here you are observing an animal cell. So this is considered as eukaryotic cell. Whereas here you are seeing a bacterial cell. So this is considered as prokaryotic cell. Here prokaryotic cell means here it is looking that it appears like a nucleus, but this is not the nucleus. So this inside the bacterial cell you find the nucleoid region. Here also you find the genetic material, as you can see the genetic material inside the nucleus. Here you are finding the nucleus, but here this is nucleoid region. So what is the difference between these two? Here the genetic material is not bounded by a nuclear membrane, whereas here the nucleus is having a definite nuclear membrane. So when the first organism was born on this earth, so those were the organisms like bacteria and blue-green algae. So these are said to be prokaryotic organisms, are consisting of cells which are said to be prokaryotic. That means here no definite nucleus, that is nucleus is not bounded by the nuclear membrane. Whereas you can see other parts like cell membrane, cell wall and so many other parts, but here the main thing is the nuclear part, which is not bounded by the nuclear membrane. Whereas here you can see a definite nuclear membrane. As the organisms got advanced, so apart from these bacteria and blue-green algae, some advanced cells were took birth on this earth, so which are said to be plant cells. And and later animal cells. So these have this genetic material that is DNA, which is bounded by a definite nuclear membrane. So these are said to be eukaryotic cells, whereas the bacterial cells are said to be prokaryotic cells.